And with that, it is my privilege to introduce Dr. Maimuna Sali Paul. Dr. Paul is a postdoctoral associate in Dr. Xiaotuan Chow's lab at the Jan and Dan Duncan Neurological Research Institute, Baylor College of Medicine in Houston, Texas. She obtained her PhD from the Banaras Hindu University in Varasani, India, where she studied regulation of notch signaling in fruit flies. As a postdoctoral associate, her research focuses on gene discovery or disease gene discovery and elucidating the mechanisms in neurodevelopmental disorders. Dr. Paul has received the National Ataxia Foundation's postdoctoral fellowship in 2022 for studying Ebf3 related cerebellar ataxias and neurodevelopmental disorders. Her long-term goal is to investigate the molecular mechanisms underlying cerebellar dysfunction in these disease conditions and translate these findings to therapeutics. And thank you so much for joining us today, Dr. Paul. I will stop my screen share and the floor is yours.
Let me share my screen. Yeah, I hope everyone can see my screen now.
Yes, we can.
Okay. Yeah, so thank you very much for the nice introduction. So today I'll be talking about a gene called Ebf3, which is a transcription factor. And I'll discuss about Ebf3 related cerebellar ataxias and neurodevelopmental disorders. So to begin with, I'll give a brief idea about ataxia for those who are very new to this field. So the people who have ataxia, they have some kind of symptoms like lack of coordination, meaning especially when they walk, and they also have speech difficulties where they have like a slow speech or slur speech that can be difficult for others to understand. And they also lack fine motor skills, meaning they have difficulties to grab or reaching to objects. They also show tremors, which is involuntary movements. And they also have gait abnormalities, which is basically an unusual walking pattern. And the ataxia people, they also have aberrant eye movement and difficulty in swallowing. So when you look into the prevalence of ataxia, around 150,000 people in the US are affected. And the lifetime prevalence rate is 50 out of 100,000, meaning these people, they had ataxia at some point of their life. And especially the prevalence rate for hereditary ataxia is like 10 out of 100,000 individuals. And when you see worldwide, 26 out of 100,000 children are affected by ataxia. Now, what causes ataxia? So the main player here is called the brain region called cerebellum, which is also known as little brain. So any damage to the cerebellum can result in ataxia. So cerebellum is a brain region that helps our body for coordination, movement, and balance. And it can affect muscle tone, motor learning, vision, and eye movement. And the recent research have shown that apart from the motor control, it can also involve in cognition. So there are basically two types of causes that you can see like in ataxia. One is acquired and another is genetic. So in acquired condition, when you take too much alcohol, it can result in ataxia. Some medicines can cause ataxia, thyroid conditions, stroke, multiple sclerosis, cerebral palsy, and even some sort of autoimmune conditions can cause ataxia. And the most recent COVID-19 has also been shown to result in ataxia in some individuals. Coming to the genetic cause, meaning there is a alteration in a genetic makeup of the individual that results in ataxia. And it could be de novo and familial. In de novo, that particular variation is present only in that individual, not in the family. But in familial, that genetic change is passed on different generations. And there are different types of ataxia that can be like genetic, like spinocerebellar ataxia, Friedreich ataxia, and ataxia telangiectasia. And there are several ataxias that are still undiagnosed. Now, like I mentioned, cerebellum is like mostly involved in the motor development. But it has also been found that But it has been also found that these cerebellum can also involve in the cognition and developments, meaning when you have a damage to the cerebellum, it is expected that other than motor functions, there are other neurodevelopmental factors can also be affected, meaning that can also cause several neurodevelopmental disorders in which basically a person have inability to reach certain cognitive, emotional, or motor developmental milestones. And these neurodevelopmental disorders include attention deficit hyperactivity disorder, autism, or learning and intellectual disability, and like a severe form of epilepsy. And it has been shown that around 15% of children in the US are affected by neurodevelopmental disorders. Now, what causes the neurodevelopmental disorders? Again, there could be different factors, like could be immune dysfunction, metabolic disorders, infectious diseases, and also genetic. So in the last 2 decades, due to the advancement in the next generation sequencing technology, a lot of genetic causes have been identified. So to date, more than 900 genes are found to be responsible for neurodevelopmental disorders. So when we study neurodevelopmental disorders, nowadays more focus has been into the cerebellum, because the developmental abnormalities of cerebellum are one of the most common structural brain malformations in the human prenatal scanning. Also, the cerebellar abnormalities are among the most frequently found in certain neurodevelopmental disorders like autism, ADHD, and dyslexia. So when you look into the image here, cerebellum has like extensive connection to the forebrain via the thalamus, which implicates its function like in different, it can affect the function of the cognitive development from different parts of the brain. And when you look into the cerebellar circuit, the key player is the Purkinje cells, which is basically inhibitory neurons. And these Purkinje cells are important for maintaining coordination and cognition. Moreover, Purkinje cells are implicated in autism presently. So in 2017, we identified several individuals who had a cerebellar abnormality in the brain MRI. So the left one is a typical brain MRI of a healthy individual, and the right one is the patients that we identified who show a cerebellar hypoplasia and a thinned corpus callosum. Later, we identified that these individuals had variants in a gene called Ebf3. And now this condition is known as Ebf3-related HADS, that stands for Hypotonia, Ataxia, and Delayed Development Syndrome. So like I mentioned, these Ebf3-related HADS individuals had ataxia and also structural brain malformations, including like cerebellar hypoplasia. Other than that, these individuals also had feeding difficulties, facial dysmorphism, preservative social behaviors, and they also show decreased pain response. They have global developmental delay and intellectual disability. They also show a speech disorder. They had autism, hypotonia, facial weakness, and genitourinary malformations. So we then looked into what this Ebf3 actually is. So Ebf3 stands for early B cell factor 3, and it belongs to the COE family of transcription factors. So in mammals, this family contains 4 members, EBF1, 2, 3, and 4. And all these EBF family members are involved in B cell differentiation, bone development, and neurogenesis. So when we looked into the protein domain of the EBF3 protein, we found that there are several functional domains. Like there is a DNA binding domain with zinc finger motif, and this domain is important for the binding to the DNA. And there is IPT domain, which function is like not yet well studied. And there is a helix loop helix domain, which is important for the homo and heterodimer formation. And the C-terminal transactivation domain is important for the transcriptional activation. And when we looked into the conservation of the EBF3 gene, we found that this gene is highly conserved across different species. In mouse, it shows 99% similarity with human. And in zebrafish, it is known as EBF3A with 96% similarity. And in fruit flies, the Ebf3 homolog is known as NOT with 70% similarity. And in worms or in C. elegans, this gene is known as UNC3 with 63% similarity. Now, when we looked into what exactly the Ebf3 does in different species, so we found that extensive amount of research has been done in the invertebrates, especially in the fruit flies. So it has been found in fruit flies that the Ebf3 homolog node can control the formation of wing vein formation. So you can see here that the panel A basically shows the control wild type adult fly wings with a distinct vein formation. In not loss of function phenotype, what we observed is that there is a fusion of the L3 and L4 vein. Moreover, there is another study reported that a loss of function of NOT can lead to decreased branching in the fruit fly sensory neurons. In panel A, basically shows the wild type larval sensory neurons, and panel B shows the not mutant sensory neurons. So you can clearly see that the branching and the arborization of this neuron has been significantly reduced. Other than these functions, NOT has also found to play an important role in wing disc development by regulating certain signaling pathways like DPP, hedgehog, and EGFR. In flies, it has been also found that NOT can regulate the blood cell progenitor maintenance and also involved in embryo patterning and neuronal cell determination. Now, coming back to vertebrate development, like in the invertebrates, it has been shown that the Ebf3 is very important, plays a key role in the vertebrate development as well. In the image here shows the developing forebrain of a mouse embryo stage 12.5, and it shows basically the strong expression of Ebf3 in the early stages of development. And several studies have shown that this Ebf3 is involved in olfactory receptor neuron projection in mice, and it can also regulate the migration of colorectal cells. And loss of function of Ebf3 can result in abnormal GABAergic interneuron migration. It is also involved in neuronal differentiation and specification, and can also regulate the differentiation of osteofibrogenic progenitors. So since this gene is highly important for development across different species, it is a potential candidate for disease, right? So we found that Ebf3 can also regulate the expression of certain genes that are involved in the cell proliferation and cell death. Nonetheless, this makes Ebf3 as a tumor suppressor gene in pediatric acute myeloid leukemia. And it's also found that the Ebf3 expression to be elevated in several cancers. Additionally, Ebf3 variants have been detected in certain cancers like melanoma, glioblastoma, and pancreatic carcinoma. So far, like whatever disease that is associated with Ebf3 was cancer. But in 2017, including our group, several others reported that rare variants in Ebf3 can cause a syndromic neurodevelopmental disorder. So once we identified several individuals with Ebf3 variants, our next step was to functionally prove or functionally implicate that the Ebf3 variants are disease-causing. So for that, for the functional studies, one can use different types of model organisms like fruit flies, mice, C. elegans, or zebrafish, or even you can use the patient-derived fibroblast. So in our lab, we used fruit flies because of a lot of advantages compared to other model organisms. Like it has a really short lifespan. It takes only 10 days to develop an adult fly from the embryonic stage. And during these stages, these are very easy for maintain, and they have a fully mapped genome. These flies are very easy to manipulate genetically, and it shows 60% identity with humans. And because of the easy manipulation of the genome, there are a wide variety of genetic toolkit are available. And these genetic tools are very frequently easily exchanged or like between the fly peers. So because of all these reasons, fruit flies are a very popular model for modeling different human disease genes. it has been found that 75% of human disease genes that are conserved in fruit flies. And there are several successful disease models have been made in flies for Alzheimer's disease, Parkinson's disease, ataxia, neuromuscular disorders, metabolic disorders, and also for cancer. So now, how we can basically use the flies to model the diseases. The biggest advantage, like I mentioned, is the technology that is available and the easiness that we can modulate their genome. So there are different ways you can engineer like the flies to model human diseases. The one way is like US Galford system. So this is a binary system that is adapted from the yeast. So this works like when the Galford binds the US, whatever in the downstream of the US get transcriptionally activated. So if you want to express your gene of interest, you can clone this into the downstream of the US. And you can express this using a tissue-specific Galford. For example, here, when you cross a fly with a wing-specific Galford and a US GFP, in the progenies will have GFP expressed in the wings. And another method is generating mutants. So you can see here that the mutant generation in flies can give a lot of details about a gene's functions. For example, this is a wild type fruit fly eye with red color. When you delete a gene called white, it results in the white eye color. Similarly, the wild type fruit fly body color is brown. When you delete a gene called yellow, the body color becomes yellow. So now, when you decide that you are going to use fruit flies as models, it is very important to recapitulate the human phenotypes in flies, right? So there are several assays that are available in flies that you can use to interpret the human phenotypes. For example, if you want to study the motor phenotypes, especially in ataxia, so one can use larval mobility assays. So here you can study or assess the locomotion of larvae, and in climbing assay, you can assess the climbing ability of adult flies. And flight assays are used to study the flight muscles related issues. And moreover, if you want to study like overall physiological relevance and also aging related diseases, you can use lifespan analysis. And for neuromuscular junction analysis, flies are very popular model organisms. Here you can study the synapse function and development. Another important tool or assay that is used in flies is the somatic clonal analysis, where you can create clones of disease cells surrounded by control cells, and you can basically study the communication between the disease cells and the control cells. So using all these techniques and assays, in the last decade, around 50 new disease genes have been discovered. So in our lab, I led a study where we identified a eukaryotic initiation factor EA4A2 as a disease gene. So we found that the individuals with EA4A2 variants had a severe brain malformations in the MRI, especially like a cerebellar hypoplasia, like exactly like we observed in the Ebf3 HADS individuals. And these patients also had thin corpus callosum. And then how we used fruit flies here to functionally validate these variants means we again use US Galford system to express the EA4A2 wild type and variants in the fly neurons. And we did a climbing assay. So we found that the EA4A2 variants have climbing defect. And when we expressed them in the wings, we found that one of the variants had a fly wing margin phenotype, which is called notching. And I was also part of a study where we identified another gene called PPFIA3, which is basically a synaptic protein. So in this patients also, they had severe developmental delay, epilepsy, and hypotonia. So since it's a synaptic protein, we used neuromuscular junction analysis here. And we found that when you express the PPFIA3 variants compared to the wild type, these had less number of boutons. And we also expressed them, the variants to study the climbing ability and the seizure susceptibility using band sensitivity. So we found that the PPFI3 variants had severe climbing defect, and also they are sensitive to the external band. Now, how we are going to study the effects of Ebf3 variants in fruit flies? So for that, we basically need to generate some reagents. First, we need to generate UAS Ebf3 lines that we did. And then we also generated a GAL4 to express the human Ebf3 wild type and the variant in the region that we wanted. So we generated a not T2A GAL4. So this GAL4 will have the expression of Ebf3 wild type and variants in the not domain. For that, using CRISPR-Cas9, we knocked in a cassette into the intron of the not gene locus. And using the recombination mediated cassette exchange, we were able to generate the not GAL4. And then our next aim was to express the variants that we identified in the patients, right? So when we did that in the wings, we found that yes, the UAS lines are nicely working, especially in the not expressing domains because we are using not T2A GAL4. The controls are the not wild type, and we express the human Ebf3 wild type and also 2 of the variants. So now we are moving to some functional assays where we are going to see like how the Ebf3 wild type or the variants can affect the fly phenotype. For that, first we need to identify the loss of function phenotype of the fly Ebf3 homolog, which is not. And that in that background, we will be expressing the human Ebf3 wild type. And next step, we will express the variant Ebf3. So to identify the not loss of function phenotype, we generated the not homozygous condition. So we found that homozygous not flies are lethal or semi-lethal. So now we are going to express the human Ebf3 wild type in this lethal background, right? So what we found interestingly that when you express the fly knot or the human Ebf3 wild type, it can significantly rescue the fly knot loss of function lethality. Whereas when you express the Ebf3 variants, it did not rescue. So this indicates that the human Ebf3 wild type is functionally conserved in the flies. It can rescue the fly loss of function lethality, whereas the Ebf3 variants did not. This shows that these Ebf3 variants are loss of function in nature. And further validate the Ebf3 variants, we did a luciferase assay to check the transcriptional activity of the variants. So we made several constructs and expressed them in the cell lines and checked the transcriptional activity. So we found that the Ebf3 wild type had a highest degree of transcriptional activity, which is expected, whereas the variants did not preserve the transcriptional activity. So this again indicate that these variants are loss of function in nature. And now we know that the Ebf3 is a disease gene, and we functionally prove that like the variants can cause pathogenic effect in the fruit flies as well. So further study this, our clinical team at the Dungan Neurological Research Institute and Texas Children's Hospital recruited more patients, and we then did a genotypic phenotypic study. So we found that around like in 40 individuals that most of the individuals with Ebf3 variants, they have the variants in the DNA binding domain and especially in the zinc finger motif. And when we analyze the prevalence and type of neurological features in this Ebf3 related HADs, we found that these individuals had delayed language milestones. They also show delayed motor milestones, and their speech was also compromised. And then we looked into the motor issues. So they also had several motor delay. Some of them had seizures. And interestingly, most of the patients, they had some interesting sensory findings that they were very, their response to severe pain was very less. Apart from this, their neuropsychiatric findings shows that they have autism or like autistic phenotypes. And like I mentioned, they also had like several structural brain anomalies, especially in the cerebellum area. So with this cohort, we wanted to, basically develop a diagnostic prediction tool to identify the EBF3 related neurodevelopmental disorders and ataxia. So we developed an algorithm basically based on the most consistently expressing phenotypes in the EBF3 HADS patients. And we scored and we give score to each of these phenotypes. So we found that you can distinctly differentiate the EBF3 related patients from the non-EBF3 neurodevelopmental disorders, because with our algorithm, the EBF3 related individuals score high. And we tested this algorithm in 2 different trials, and 2 of these trials, it gave a significant results. So this will help to differentiate basically the EBF3 related neurodevelopmental disorders from others. And then we also developed another algorithm to study the domain severity. So we found that those variants in the coding zinc finger domain had the highest developmental delay scores compared to the other regions. We also did this in 2 different cohorts, 2 different trials to see the recapitulation. So we found that in 2 of these cases, it did recapitulate the same results. Now we did apply this algorithm to understand the distribution of this score in the individual. So we found that those variants in the zinc finger domain always had the highest DBS score. And also, when you look into the developmental delay severity scores, those variants, those patients with the variants in the zinc finger domain will basically clustered in the zinc finger domain. So this analysis basically shows that the EBF3 HADS individuals with the variants in the DNA binding domain, especially in the zinc finger motif, they'll have the most severe phenotypes. And we also confirm this domain-specific severity using Fly loss of function assay. So we basically repeated the loss of function assays with the lethality phenotype. So we found that, like we did earlier, the EBF3 wild type can significantly rescue the lethality, whereas those variants in the zinc finger motif did not. Interestingly, the variant in the DNA binding domain, but the outside the zinc finger motif had a significant lower risk efficiency. So this again proves that the variants in the zinc finger domain have is a strong loss of function allele compared to those in the non-zinc finger area. We also did the luciferase assay to check the transcriptional activation. So we found that yes, the variants in the zinc finger domain have the lowest transcriptional activity, whereas the variants outside the zinc finger domain, their transcriptional activity was conserved just like the wild type. So now we know that like a EBF3 acid disease gene, it can cause like severe phenotypes. How we are going to basically use fruit flies to understand the mechanisms behind EBF3 related HADS. So for that, we are again going to express EBF3 wild type and the variants using UAS-Galford system. So we are going to express EBF3 wild type and variants using not T2A Gal4 in the wings. So this is like the fruit fly larval 13-star wings. And these larval wings, when they develop into the adults, we found some interesting phenotypes. So here, the first wing is a control wing with a distinct venation formation. And the control, where there is no UAS lines, just the not T2A Gal4, it was similar like the wild type wing. Whereas when you express the human EBF3 wild type in the non T2A Gal4, we found that there is a loss of anterior crossvein, and the distance between the L3 and L4 was also significantly increased. Interestingly, when you express the EBF3 variant, we found that the wing vein fusion phenotype in the anterior crossvein region, and the distance between the L3 and L4 was also significantly reduced. So now we thought that like how we can basically use these phenotypes to identify some EBF3 modifiers. So we looked into the literature and we found that there's a group that did a genome-wide mapping for not binding sites. And they identified several genes that have the NOT binding sites, meaning those genes will be regulated by NOT. And these could be a group of genes that could be like can regulate transcription, axon guidance, nervous system development, wing development, or muscle development. So we leveraged that data and we planned a screening to identify modifiers of the Ebf3 phenotypes from these genes. So we basically selected some candidate genes with at least 2 NOT binding sites. And we grouped them into different types, like those who are affecting wing development, muscle development, and neuron development. And we obtained RNA lines, which will help to knock down this gene's function. And we express them in the background of human Ebf3 wild type overexpression or the variant. So this is my cross scheme to knock down the candidate gene. So first we have a control where we'll have the expression of RNAi with a not T2A gal4. And then we will be knocking down the candidate gene in the background of US EBF3 wild type expression, and then in the background of US EBF3 variant expression. So this is the control phenotype that we are going to use in the screening to see if these can be modified, right? So the first candidate that I used is CI, which is cubitus interruptus. So when we expressed this RNA line to knock down the gene cubitus interruptus, we found that there is not much significant difference in the background of EBF3 wild type, like these two wings are almost similar. But interestingly, when this gene is knocked down in the EBF3 variant overexpression, we found that the wing-vein phenotype, wing-vein fusion phenotype is worsened. And then we next knocked down a gene called GPRK2. So when we knock down this GPRK2, we found some interesting phenotype in the EBF3 overexpression. There is extra vein material. And again, in the EBF3 variant overexpression background, when you knock down the GPRK2 gene, we found that the wing fusion phenotype is worsened. And then I used another gene, which is mirror. When you knock down the mirror gene in the background of EBF3 variant, we found that you can basically rescue the wing-vein fusion phenotype. And another gene, we glued, when you knock down, we found that it can rescue basically the EBF3 wild type expression, wild type phenotype. Like here, you don't even see the anterior crossvein, but when you knock down the V-glut RNA, this is restored. And also the EBF3 variant phenotype was also rescued. And similarly, I knocked down a gene called Pimilio, and I found that in the background of EBF3 variant overexpression, it can significantly rescue the wing-vein fusion phenotype. So to summarize my screening, I basically found 5 genes that can, when you knock down this gene, it can modify the EBF3 vein fusion phenotype. Cubitus interruptus, which is basically a sonic hedgehog pathway effector, and it can worsen the phenotype, and the mouse homologous GLI family of transmission factors. And then another gene is GPRK2, which can also worsen the wing-vein phenotype. Which is the mouse homologous GRK family. This can also regulate the sonic hedgehog pathway. And other 3 genes which are basically rescued the wing-vein fusion phenotype were mirror, which is involved in the neuron maturation, and V-glut, which is involved in the uptake of glutamate into the synaptic vesicles, and Pimilio, which is involved in neuronal excitability. And interestingly, the Pimilio gene is also implicated in spinocerebellar ataxia, and the cubitus interruptus also implicated in a disease called post-axial polydactylity type A8. And we also looked whether these mouse homologs of the genes that we tested in the flies have expression in the cerebellum. So we found that yes, most of the genes are highly expressed in the mouse cerebellum as well. So this indicates that like these genes could be seriously affect the EBF3 function in the mouse as well, and it can basically regulate the EBF3 expression. So to summarize my study, we basically identified EBF3 variants can cause ataxia and neurodevelopmental phenotypes in affected individuals. And we found that EBF3 is an essential gene for survival, and it is involved in neurodevelopment and invertebrates and also in vertebrates. Our food fly model shows that EBF3 variants are loss of function in nature. And our phenotype-genotype study shows that the EBF3 HADS individuals shows that the variants are commonly found in the DNA binding domain, especially in the zinc finger motifs, are associated with severe phenotypes. Our screening study shows that the not target genes in food flies identified several modifiers of EBF3 phenotype. So in the future, directors are we are planning to extend the study in a cross-species approaches where we will be using fruit flies to identify the molecular and cellular pathways, and we'll be using mouse to understand the behavioral and anatomical perspective of the EBF related HADs. So with this, I would like to thank my mentor, Dr. Chow, for all the support and my lab members. And I also would like to especially thank National Ataxia Foundation for the fellowship. Also would like to thank other funders to our lab and our families and collaborators for making this study a success. So yeah, thank you.
Thank you for a fantastic presentation, Dr. Paul. We're now going to be opening up the floor for questions. As a reminder for attendees, if you'd like to ask a question, you can type it into the Q&A box at the bottom of your screen, or if you'd like to ask your question verbally, you can use the raise hand feature and we will be able to unmute you so that you are able to share. And while folks are thinking about their questions and perhaps typing away into the question box, I wanted to open up with a question of my own. At the beginning of your presentation, you mentioned that EBF3 is a tumor suppressor gene. We know that there's certain kinds of ataxia, such as ataxia telangiectasia, where there's increased cancer risk since ATM is involved as a tumor suppressor gene, as well as connected to ataxia pathways. Is there anything known for folks who have EBF3 related HADs?
So far, in our patients, we haven't seen cancer, but we have also noticed that some of the EBF3 variants are common in the cancer and also in the HAD. So since this is the EBF3 variants are de novo, like germline. So it could be possible that maybe in their lifetime, at some point of time, they might develop some kind of skin cancers like melanoma. Yeah.
It could be later in life. That makes a lot
Yeah.
of sense. And then out of personal curiosity, is there overlap in those areas of the gene where the mutations relating to cancer versus the areas related to HADs are? Are they both within that zinc finger domain, or are they clustering in different regions of the gene?
So mostly the cancer variants and the EBF3 HADS variants are different domains, but there are very few variants are overlapped.
Well, thank you so much for clarifying. And going through another question that I had. So you mentioned early on when you were doing these preliminary fly data that you found that homozygous loss of NOT was lethal. Do you have any hypothesis of why this could be? Is it about the essentiality of the gene, or is there something else going on here?
Yeah, so this gene has been shown to be important for the embryonic pattern formation. So in fruit fly, that is the earliest stages of development. So that I think that will be the reason. If you have not a proper embryo formed, then it's difficult to further, development. Yeah.
Well, that makes the rescue of that phenotype all the more impressive.
Yes, exactly. Yeah.
And then another question I had as we're waiting for some to come in for participants, shifting from your fly model work to some of the more clinical work you did with human participants. It was very fascinating, the diagnostic model that you put together. Could you share with us a little bit about how that could be implemented in a clinic now, and what was the onus of creating such a system for to be used?
Yeah, so when you see the phenotypes of the neurodevelopmental disorders, I have worked on 2 other genes like EF4H and EPPF5A3. Although the genes are different, their phenotypes could be very similar. So it's ataxia is like very common, and developmental delay is very common. Epilepsy can be like common. So it's important to recruit more patients and to identify. It'll be nice to differentiate between these gene specific phenotypes. So like when you develop such algorithms, like a clinician can easily differentiate, oh, this is ABF3 related neurodevelopmental ataxia, and this is not. Yeah.
Then how would that information be used in clinic? Would it be for for diagnosis, there's a
Yeah,
clear
for diagnosis.
there, but anything later on in terms of treatment or research participation, how could that be helpful?
Yeah, so
How would
this
that
is
information be helpful for patients and family
yeah, so this will
members?
be like definitely helpful for diagnosis, and then also for further recruiting like more individuals to the study, which helps for the further therapeutic help. Yeah,
As more participants, more data, we're able to figure
yeah,
things
yeah,
out a
yeah.
little bit easier. And then sort of jumping back to the gene itself, EBF3, it's fascinating that these mutations seem to be clustering around this zinc
Mhm.
finger binding domain.
Yeah.
Any hypothesis of why seems to be the critical area for this gene and this protein to be causing this cascade of symptoms down the line?
Yeah, so we're not completely sure, but yeah, definitely like the zinc finger motif is like the pivotal for the DNA binding domain. So without that, like ABF3 cannot act as a transmission factor. So maybe that's why it's like more susceptible to this kind of mutations. And though that particular area is also like highly conserved across different species as well. So there could be some reasons that like evolutionarily that area is like conserved. But yeah, like why these mutations are like exactly hitting there? Like not sure. Yeah.
A little bit more time to tease that apart. It's probably very complex in terms of the factors going into this, but
Yeah.
clearly you're showing how important this area is if it's
Hmm.
conserved across so many different species.
Yeah. Yeah.
And then Um, shifting gears a little bit, I noticed in the graphs you had, oftentimes you were stratifying between male and female participants.
Hmm.
Could you speak a little bit for folks on the call about why you do that within your data set? And did you see any gender differences within your
Yeah.
population?
Yeah. So usually, like some of the neurodevelopmental disorders, like autism and ADHD, dyslexia, it's very common in males rather than females. So we don't know like the exact reason behind that. So we also wanted to see like in EBF3 related neurodevelopmental disorders whether this is the case. But so far we haven't noticed any gender sex specific differences. But yeah, our sample size is like very low. Also like yeah.
It's interesting you're doing that up front so that if any sex-specific differences
Mhm.
occur later on, that you'll be able to see those in real time.
Yeah.
So fantastic. Jumping a little ahead in your presentation, at the end, you mentioned the Panimula 1 gene and how this rescues phenotype, and this is connected to SCA487. Have you
Mhm.
teased this apart any more in terms of figuring out or connecting this or exploring this connection in more detail? It's fascinating to see two forms of ataxia being connected this way.
Yeah. So there is always chance that like although different types of ataxia, maybe the pathways are similar, but like the how Pumelio is affecting not is not yet been studied. So we are, trying to get reagents and like make reagents and trying to identify the molecular and cellular pathways behind that. That could be like maybe like a common pathway, but maybe there's a minor difference can result into two different types of ataxia.
Of course. And then one last question from me for any young graduate students or early career postdocs on the call who are interested in going to ataxia research or neurodevelopmental research like this. Do you have any advice for them getting started or continuing in this career path?
Well, yeah, I mean, I would encourage them to, definitely come to this field because although these are like very rare conditions, but there are like when you look into that, like there are several people who have like suffering from this, and definitely the students they should reach out into like different labs or can attend conferences, some like webinars like this to get the idea that like what has been done and what there are so many things can done in future also. So yeah, I would like encourage them to continue to this.
Well, thank you so much, Dr. Paul. I'm not seeing any other questions in the chat, but we really appreciate you sharing your research with us today and taking the time to talk with us about what you're doing, what you have done, and what you're going to be doing in the future. So thank you so much for your time and for your expertise.
Thank you. Thank you.
And thank you to everyone on the call who stuck out with us for this hour to learn a little bit more about some research that's being done on neurodevelopmental conditions and for ataxia. We very much appreciate your time and hope you have a wonderful day ahead. Take care, folks.